Good afternoon. Now it's 1 o'clock, and today we are going to hear some very interesting interviews with Kåre Enkilde, the CEO of Amniotics, and Sandra Linster, scientist and professor within pulmonary diseases, and especially lung transplantation. Then we deep dive into lung transplantation and try to build a bridge between the science world and Amniotics as a company. We would like to say hello to the audience as well, and just some practical issues. If you want to post questions either to Kåre or Sandra during the presentation, please post the questions in your lower right corner, and I will forward the questions to Kåre and Sandra. And with those words, I would like to hand over the mic, or we'll hand over to Kåre from Amniotics. Welcome, Kåre.
Thanks, Klaus, and I'll do my best to virtually grab that phone and start getting into motion here. So we don't have a lot of time, so I'll just do a very short presentation of Amniotics. Not sure if you've heard too much of us, though we're trying, of course, doing the best we can to show who we are and what we come from. If we just very briefly touch upon who we are based at Lund, southern Sweden, based at the old AstraZeneca site. We have built a full platform of mesenchymal stem cells taken from amniotic fluid, hence the name. We were listed on the First North Nasdaq just this summer, and now we're building a pipeline of tissue-specific mesenchymal stem cells from the amniotic fluid. And that might be interesting to understand that the amniotic fluid is surrounding the child, the neonate during pregnancy, and hence it carries all the cells and ingredients, both of course from the amniotic membrane, from the maternal side, but also from the child's side. Not touch upon a lot of this, just pushing again that we're working with these tissue-specific cells, meaning that we're targeting cells for the same indication, or more or less tailoring the cell. So you could say that we're using lung-specific cells for treating lung diseases. Likewise with kidney, we're taking kidney-specific cells for kidney diseases and skin and so forth. And that's really, you could say, to build a broad pipeline of products based on these cell and specific cells, and creating that stronghold as a company and building a broad pipeline. As you also alluded to, Klaus, earlier on in our start here, before we went live, we've built a full manufacturing capability enabling us to produce these cells in-house and having them GMP ready. Also understanding that working with cell therapies, there's also always a discussion about scalability. How much can you scale compared to small molecules and antibody? That's one of the reasons, of course, targeting orphan indication and rare indication. But it's also important to understand that our products generally can treat more than 1,000 numbers of patients. So you could say a single donation of amniotic fluid, a single pregnant woman that will consent to let us source amniotic fluid will carry enough for more than 1,000 treatments, 1,000 patients. So there is certainly a lot of scalability in this setup. And also understanding, I touched a bit upon early on the different indications that we're looking at. We're looking at the neuronal indication, we're looking at the kidney, at the respiratory and the dermatology. And all of these areas are really looking to grow quite a lot in the coming years. And a lot of that is also, of course, driven by age. If we look at CNS diseases, a lot of coming from Alzheimer's, Parkinson's, and so forth. A lot of the same when it comes to kidney, where we have diabetes-induced kidney diseases, we have cisplatin-induced kidney diseases, and so forth. And the same goes with respiratory indications. Some of these respiratory indications also are due to an aging body, and the same goes for skin. This is the overview of pipeline products, where we are. We're looking to have a first patient in, that what we're targeting within COVID-19 hospitalized patients, first half this year. So we're really working on that tightly. We're also looking at idiopathic pulmonary fibrosis, and as we're talking about lung transplantation, because this is really areas that kind of combines into each other, because acute respiratory distress syndrome can lead to lung transplantation in the end stage. Likewise, can idiopathic pulmonary fibrosis. So these are indications that can lead to lung transplantation in the end. And from there, we'll start to broadening the pipeline into some of the other therapy products. This, you could say, is a quick overview slide of the product, how we work. We have a device to collect the amniotic fluid from planned C-section, so it's really safe and without any ethical issues. It's a source material that's normally just medical waste. Then that gets transported to our facility where we work with the product, select the product, expand the product, so we have all the way to fill and finish, ready for clinical trials, and moving forward with that. So really understanding that we are controlling the full value chain and the full process chain, all the way from the fluid and all the way to fill and finish, to having it arrives ready for clinical trials, which I believe is a very stronghold when we're thinking about cell therapy, advanced therapy, medical products. So this I'll not dwell a lot too into. We don't have a lot of time, but as mentioned, we really have the capability to produce the products ready for clinical trials. We built a rather strong GMP facility for that. We have in-house QC labs, we have inside storage facilities, we have manufacturing rights for our products and so forth. So really having a stronghold on the regulatory side on this as well. As mentioned earlier on, we're targeting first half this year to take in the first hospitalized patients with COVID-19. Really looking at some of the, of course, safety and tolerability of Pulmostem product, but also looking at some of the early efficacy markers, looking at TNF alpha, IL-6, TGF beta, and some of those, both you could say on the inflammation part, but also moving into the fibrotic areas such as Krebs von den Lungen 6 and so forth, some of the markers for fibrosis diseases, and also looking at fatigue. We all know that this post-COVID syndrome has a tendency to kind of reduce energy levels. So that is also some of the areas that we will target with a questionnaire. And just to understand, you could say normally when you look at a small molecule and antibody, you're very particular on what the indication are. But here we're working with a cell product that has several mode of actions. And that leads to the fact that this can be used in several different indications, such as you could say this is a paper in a journal looking at this could potentially also be used in pulmonary hypertension or chronic pulmonary diseases or bronchopulmonary distress. So in a lot of indication this can go into. Just a quick overview. Cell therapy is still a nascent market. That's of course important to understand, but it's also looking to grow quite rapidly because we have an aging body, we have oncology and so forth. So really moving forward as we see it. Also, again, pushing on the fact that we are working with tissue-specific cells from neonates. So very potent cells with a tissue specificity for that organ. And we have a broad pipeline targeting these very different applications. We also brought in a number of people with a strong capabilities, both from pharma and medtech and so forth, really enabling us to build the company and push forward with this, really enabling us to execute on what we believe in. And with that, I'll just end it over because I think what will be very important here is also to listen more to Sandra and her experience with lung transplantation. What's the needs here? Because this is really a high need area.
Yes, thanks a lot, Kåre. And just before we hand over the mic to Sandra, I have one question that leads to Sandra actually, because you entered into this collaboration with CAMP. And could you very shortly describe CAMP for us and the benefits of enter this facility, Kåre?
Certainly, you could say Sweden really has a stronghold when it comes to advanced therapy medical product, and advanced therapy medical product carries both the area within gene therapy and cell therapy, and Sweden really has a strong area and strong foothold in this. So CAMP is Center for Advanced Therapy Medical Products, without the T, so it becomes CAMP, Center for Advanced Therapy Medical Products, and it's a Vinnova funded project, and that brings together the national capabilities within Sweden. So you're able to work together with other companies, with academias and so forth, bringing products all the way from preclinic and all the way to clinic, and with all those challenges that may be both on the CMC part, the fill and finish part, the production, and so forth.
Good. Thanks a lot, Kåre. And with those words, Sandra, I would like to welcome you, and I just have to change the presentation here, 2 seconds.
Thank you very much.
The word is yours, Sandra. Welcome.
Thank you. Thank you very much. So, yes, so my name is Sandra Linster. I'm a cardiothoracic surgeon and also professor in cardiothoracic surgery. I also do a lot of research within the field of lung transplantation. So lung transplantation and what problems are we facing? So, well, the major obstacles in lung transplantation is actually the imbalance between the demand and the supply of donor organs or suitable donor organs. Because many of the donor organs that we receive, we have to turn down because they do have an injury that we today cannot handle. And because of this scarcity of donor organs, there are deaths on the waiting list for lung transplantation. But we also have problems after the transplantation. So many patients do not tolerate the new lung. And the survival after lung transplantation is only 50% after 5 years. So we have a quite problematic situation. So due to the lack of organs, attempts are being made to find ways to restore the function of discarded lungs for transplantation. So what are those discarded lungs and what happened to those? Well, previously healthy lungs, they can be damaged within the donor. And within a donor, they often develop acute lung injury. And that can be secondary to aspiration, infection, or actually just due to brain injury. So these lungs are lungs that we cannot use today, and those lungs are the lungs that we turn down. So only 20% of the donor lungs we are offered, we can use today for transplantation. So that doesn't really cover our needs. So in only Sweden today, there are 2,000 people or patients that do have a respiratory failure and that would need a lung transplantation. But due to the scarcity of donor organs, they don't even make it to the transplant list. So in Sweden, and the situation is similar in Denmark and all over the world. So only in Sweden, we do 50 lung transplantations a year. So who should we choose? And who should we not choose? This is a big problem for us. The COVID pandemic has also been very problematic for us because we see a lot of late complications with severe respiratory failure, leading to such a severity so they do need a lung transplantation. So the major problems in lung transplantation is the urgent need of more suitable donor organs to cover the needs of what actually is needed. But there's also an urgent need of improving the results after transplantation. So these are the two major obstacles we fight with in transplantation. And this collaboration together with Amniotics, we strive to both increase the suitable donor organs, but also to improve the results after lung transplantation. Thank you.
You're welcome, Sandra. So when you talk, could you elaborate a little on this primary graft dysfunction when we talk about lung transplantation? Because that's the rejection that the patient experiences. Yeah?
It's a good question. Well, it's primary graft dysfunction is where the lung does not function properly during the first 3 days after transplantation. And that is sort of an intolerance to the new organ. And we know when the patient develops primary graft dysfunction, we know that they have a higher risk of chronic reaction. So chronic reaction is different to primary graft dysfunction, and chronic reaction is something that develops over time. But we know that it has a strong relation to primary graft dysfunction. And that's why it's so important for us to treat primary graft dysfunction. Today, we don't have any treatments for primary graft dysfunction. So that's why it's so interesting for us.
Interesting. And can you link that to the collaboration within CAMP and with Course Company? Are you able, with the stem cell therapy, to elevate this condition? Maybe it's too early, but if you could answer that question, Sandra.
Yeah. So that is what we are trying to do, and let's see if we succeed. So we are in this collaboration with CAMP. We both try to treat the lungs prior to transplantation to make not suitable organs become suitable. But when we have transplanted those organs, we also try to treat within the recipient, within the patient. So we have two targets here, both to increase the donor organs and also to increase, to treat primary graft dysfunction post-transplantation.
So that's both short and long term, as I understand you. Both in the short term, so it doesn't reject the organ, but also in the long term, so the patient experience a better life quality and less, maybe not hospitalized as much as you will usually see.
Yeah. Yes, that's correct. I mean, the big problem for us is actually that it's the mortality is so high.
Yeah. Okay. Yeah. Well, that's even worse.
Yes, that's even worse.
Yeah. Yeah, because it's quite complicated. Cole mentioned for me, I think a couple of months ago. So one thing is to do these lung transplantations, but all the preparation and the service afterwards is really demanding for the system and for the patient as well. With the current treatment. Yeah.
Yes, that's correct.
Yeah, and that's probably one of the end goals you would like to examine as well. So with the amniotic fluid and Pulmostem, are you able to improve this? I could imagine.
We hope so.
Yeah, that is certainly what we hope for. That is really what we're targeting. That's really what we hope to deliver.
Yeah. Could I ask a question about the indication arts? And maybe ask you first, Sandra, and then you could answer the question afterwards, Kåre. But could you describe to us, to the audience, in a very simple way, how Pulmostem might help patients who suffered severe lung damage as a result of the COVID-19 infection?
So within this project, we work with lung transplantation, but what we had experienced in my field, in my clinical field, that we today have more patients under investigation for lung transplantation or at the waiting list that need a lung transplant due to the late causes of COVID-19.
Thanks a lot. Yeah, sorry, Sandra. I know you're an expert within transplantations, but on the other hand, in your presentation, you linked severe post-COVID-19 with this project as well. So that's why I asked you. But maybe, Kåre, you know a lot about this, I know.
I would, you could say, strengthen what Sandra says. Generally speaking, you could say across the world, we see an increase in double lung transplantation, which is most likely due to COVID. It's of course sometimes difficult to say exactly what's causing that, but quite what can be seen, and normally you wouldn't see that increase, such a high increase in the need for double lung transplantation if it wasn't due to the infection with SARS-CoV-2 leading to COVID-19 and so forth. And COVID-19, you could say, is also a kind of acute respiratory distress syndrome covering that it can be caused by several different things. For COVID-19, we just know that it's SARS-CoV-2 because we did the qPCR earlier on, and we know that that's the infection that drove that development. I hope that kind of brings a little understanding in. If I should speak to how the product works, it works in several different parameters, and hopefully you could say that's part of doing a clinical trial. That's really to get more grasp on what is that in a human setting. But one of the things is, of course, reducing the inflammation. We know that some of the inflammatory products are potentially causing damage to the lungs. We also can see that some of the corticosteroid or dexamethasones has been used in COVID-19, but we also know that using that type of product is causing damage to patients. So it's not necessarily a good thing to use a lot of that. What you often want to do is a product that is kind of better at mingling that out and setting the stage for that. And that's really also where we see a stronghold for our product, because it works on many different areas and not just without or within anti-inflammation. It can also reduce some of the fibrosis markers and so forth.
Thanks a lot. So if we go a little back to the lung transplantation area and your cells core, how would you deliver the cells to the patient? Once, can you tell us a little in a simple way how? So you have this patient with an injury, and you have the short and long term prospect. We already covered that. But are you going to be present at the hospital, or are you delivering it to Sandra before, or can you tell us a little about the logistic?
Logistically wise, it's a frozen product. Very cold, but we already know how to handle cold chains due to the RNA vaccines that we already see. So we know how to handle cold chains already. So that's not a big problem. So we will deliver vials, frozen vials, to the hospital, and then the hospital's pharmacy will be sending that out to the clinicians doing the surgery or.
Yeah, good. Well, then I can ask the audience if they have any questions for Kåre or for Sandra. Then you are more than welcome to post them on your lower right corner of the screen, and I will forward them to the presenters. There's no questions right now. Let me see if I can find some more questions. For sure I can. Kåre, now you're targeting amniotic fluid, and maybe there's different kinds of cell-based immunotherapy. What advantages have you experienced with your cells? Because you of course talk with your colleagues and so on and so forth, compared to other kinds of cell-based immunotherapy, because this is a very interesting area in general.
It certainly is, and it is an area that can be difficult and hard to fully grasp, because there's so much ongoing and a lot of development in that field. If we should, you could say, try to focus on the stem cells and the mesenchymal stem cells, there's generally speaking focus on either using bone marrow derived mesenchymal stem cells or fatty tissue mesenchymal stem cells taken from liposuction, or some of the other you could say birth associated material. In that sense, I would say that working with amniotic fluid is, for one, we have it in a fluid. All the other things are in a tissue where you need to extract it, which by itself can cause damage to the cells. Here we're working with them already in a fluid. Secondly, these cells, you could say, we do the selection, we do the sorting, we do work with them. So we are working with the neonatal, so the potent, the young cells from the child. We're also working towards having the tissue-specific cells, which few companies are working on. There's a general understanding in the field of mesenchymal stem cells. There's a need to kind of work around some of this. But I would say working with tissue-specific cells enables us as we see it, potentially to deliver better therapy, because these cells have already, you could say, been in contact with that tissue, and the tissue kind of imprints epigenetics in these cells, enabling them to that target or to that tissue. Then there's some of the, you could say, other areas. These cells are smaller than what we see from some of our, you could say, colleagues in the field, enabling them to better, you could say, cross or come around in the body through the veins, through the arteries, and so forth.
Thanks a lot, Kåre. Well, time is already up. But Sandra, do you have anything to add here in the end that we should follow, or?
Broad question. I would say that no, I think it's a very interesting field, and I do think that we have high hopes that this will have an impact on the lung transplantation field. So I'm very I'm looking forward to this project together with Amniotics.
Thanks a lot. And what can we do to get more lungs, more donor lungs? Is it a matter of signing up, or maybe it's a funny question, but I see from the demand-supply balance, it's really demanding.
Yes. Well, the first thing I was, well, of course, signing up for donation is, of course, important. But since 80% of the donor lungs we cannot use them today for transplantation. So because of they have an acute lung injury, by being able to treat those lungs prior to transplantation and make those lungs suitable for transplantation, that's how we increase the availability of suitable donor organs.
Yes, thanks a lot. Well, with that said, I will thanks a lot, Sandra, for participating in this event, and we hope that we can invite you another time. And as always, Kåre, thanks a lot for the presentation, and to the audience, thanks a lot. And we will, of course, post the video afterwards on our YouTube channel, and then we look forward to see the company soon again. Thanks a lot, Kåre and Sandra.
Thanks, Lars.
Yeah.